This is Dr. Rindy. I'm reviewing a paper that I read that was very helpful in understanding some of the differences between stool testing. So we're looking in this paper at a specific population of people who have been tested positive for clostridium difficile, and that's a diarrheal illness. They were compared to a population of people who were tested negative. These stool samples were used in these people who have already been detected. They took the sample and started to run the samples into other technologies like 16S RNA sequencing and MSS, which is metagenomic shotgun sequencing. 16S rRNA and metagenomic shotgun sequencing are some of the more commercially available microbiome detection systems that are being used. So just before we get into the discussion, just to let you know the consistency of the data in the samples that were used. Clostridium difficile was detected in 90.9% of the C. diff positive samples in the 16S RNA gene sequencing, and C. diff was detected in 86.3% of the C. diff positive samples using metagenomic shotgun sequencing. And PCR analysis correlated with the abundance of C. difficile that was reported in both of these technologies. So we're just going to go into some background, and sorry for the little typo there. The advantage of whole genome sequencing compared to culturing isolates is the metagenomics can assess communities of organisms, but we don't have to isolate an organism to study it. And they can do this with a specific gene such as 16S RNA, or by this metagenomic shotgun sequencing using total nucleic acids within the sample, such as a stool sample. And one of the things that's really key is 16S rRNA gene sequencing is limited to the phylum and genus level of specificity, and that is potentially a problem. So when you're getting your data back from someone like Thrive, we're getting this data on the phylum and genus level, which is probably better for population shifts, epidemiology, public health studying of the microbiome, and understanding kind of global aspects. But when you're talking about disease or specificity, it might not be as helpful. So it should be pointed out that it is very expensive to run these tests, the MSS tests, compared to the typical PCR or cultural test. So that is one thing. But the thing that is really advantageous is the MSS can detect additional viruses, fungi, and other microbial components. And like we said, helminths, worms that could not be cultured otherwise with traditional testing methods, just because some of these organisms are anaerobic and also won't grow on a culture. So because dysbiosis is an issue of not just bacteria, but a lot of bacterial and microbial components, it's probably best using MSS for understanding the totality of what we're dealing with in dysbiosis. One of the other things that's really interesting is that when you're doing these testing, you often see other pathogens arise. So people in healthy populations may have levels of potential pathogens like Staph aureus, Candida albicans, which at the time might not be bothering them, but if they do become immune compromised, such as get a illness or go into a different type of medication that causes immune suppression, or their health changes, these populations, if they're residing, may come to life and cause symptoms. So I think it's important that some of this testing can be used for prevention to find future threats, and maybe is a good way to look at this is if the person is not symptomatic and they're doing this as screening, is to potentially inhibit some of these or downregulate some potential pathogens, so that they can not be hindered by these pathogens if they were to become immunosuppressed. The other thing that's really important in disease that we're learning is that there's a concept of co-infections, because in say like diarrheal illness or some type of digestive disorder, if we're just looking for one particular cause, for example, Giardia, and we're not doing a wider cast of looking for co-infections, we may just be treating the wrong bug if we decide to deductively say that if we just test for Giardia and we find it and we treat it, well, if the patient doesn't get better, does that mean that the Giardia was not cleared? Well, let's say it was cleared and you actually had another issue like an overgrowth of Candida albicans that was contributing to the diarrhea, but the whole condition was treated with an antibiotic, making the Candida albicans worse. So understanding the totality of the different organisms that are in abundance or present can help with strategizing more effective treatments. So when we look at MSS, you can see that it can go down to the species level. So like you can see all the way down to the actual name of the species. Now this is really important because as research is coming out on IBD, IBS, other conditions like arthritis and rheumatologic conditions, we're looking at specific species to identify if they're present in these populations in abundance or they're deficient. And so this helps us with understanding and being able to detect things at this level. So in this study, there was one patient who had 35.5% Candida species in their colon, but they were diarrheal, but they were negative for C. diff, and it was found out in retrospect that this patient was treated for months with many antibiotics, and so they were able to detect that perhaps the Candida was a cause of the diarrhea or a consequence of antibiotic diarrhea. So I'm just pointing out, again, I talk about Saccharomyces boulardii being important to take with antibiotics. This sort of backed that up even further. So again, the study just really talked about how you could use the Metagenomics shotgun sequencing for rare and unexpected viruses or in a non-responding person. However, one of the things that was interesting is that accurate detection and identification also depends on the reference database that the lab or the system is using. And I'm looking into this further. I figured there'd be one universal reference database that everybody uses, but apparently there might be some variety of databases. This reminds me of sort of like if you're using a word processor, and some word processors will recognize certain vocabulary and others will not, even though so if you were to make an error in a document, there might be something that flags and that's not recognized by whatever database that you're using. And what I don't know is why there's variety in the databases that they're using on these technologies. It seems to be concerning to me. And so you could be using a system that and the microorganisms might sail by undetected or unrecognized. That might be significant. Likewise, there might be an error in the detection of the organisms, because basically the way DNA works is there's a bunch of codes, and there's a code that comes out with different sequences of nucleotides, and that's matched with a known sequence in a microorganisms. So there can be some mismatch or some overlooking of microorganisms based on whatever is being used as the reference database. So the other thing that's pointed out in this paper is that, like, let's say we get a report with all these different microorganisms, we still don't know who is causing the manifestation of the disease. So it still leaves some concern as to like what we treat. If you get 5 or 6 or 7 different bad guys in a lineup, then how do you detect which one is responsible for the crime? There has to be some trial and error usually, and that's similar to this. Is that we have to figure out if there's like another line of detection if you have 7 suspects. But most of the time at that point, you just have to sort of treat clinically and see if we can make our best guess. But this is a far more specific approach than I've seen, traditionally. So the takeaway is that, one thing I would say is there is another approach called multiplex PCR, and what this does is can sequence on a very deep level organisms that we're trying to find. So let's say that there's 12 different microorganisms that we know can cause diarrheal illness. You can take a stool sample and just sequence out just those specific 12 microorganisms and look for them in a clinical scenario. This seems also a very practical way for looking at detection of causative agents of diarrheal illness. Again, it's going to leave out rare or unknown causes. If you have an idea of what you're looking for going into a study, then you're going to either find those things or not, but you're not leaving open the possibility of finding other organisms that might be involved. So that is another way to go with this. And so the takeaway is, should we even use 16S RNA in a clinical setting? I don't know. It's questionable, how helpful that is. But I definitely think the metagenomic shotgun sequencing has a lot of potential of application. And then the multiplex PCR can really be more specific here and now application of things that we know from the research, and we can kind of zero in on those things to keep ourselves clinically accurate and not get too lost in perhaps irrelevant data. So the question is, do we cast a wider net or a narrow net? Do we do both? Do we start with more of a narrow net with something like multiplex PCR. And yeah, I think this is getting us to a point of more personalized treatment, if anything. So thanks for tuning in, and hope you enjoyed this review.